Right now, your intestines house about 100 million neurons, roughly 4 times the number in your entire spinal cord. This dense network lies in 2 layers called the enteric and submucosal plexus, which coordinate every contraction and secretion. The sheer volume rivals a small city of nerve cells, and it operates without any input from your brain. Imagine a traffic system that runs flawlessly even when the central command center is offline. Your gut's neural traffic lights fire constantly, keeping digestion moving. The enteric plexus alone contains around 70 million neurons, enough to outnumber the spinal cord's 30 million by a wide margin. This hidden powerhouse explains why you can feel nausea before your brain even registers a smell. Your stomach acid, strong enough to dissolve a nail, is regulated by these same neurons, showing how chemistry and circuitry intertwine. The enteric nervous system evolved before the brain, proving that gut intelligence is ancient and essential. Think of your gut as a self-driving car, while the brain is the traffic controller. The car's onboard computer, your enteric nervous system, processes road conditions, adjusts speed, and brakes without waiting for signals from headquarters. This analogy works because the enteric plexus monitors stretch, while the submucosal plexus senses chemical composition, together forming a closed-loop control system. Just as a car uses sensors to detect obstacles, gut neurons use mechanoreceptors like Pieto2 to feel pressure and chemoreceptors such as GPR41 to detect short-chain fatty acids. The system then releases neurotransmitters, for example acetylcholine to stimulate muscle contraction and nitric oxide to relax it. Because the gut can respond in milliseconds, you can feel a sudden cramp before you even think about it. This autonomous operation is why you can vomit while unconscious. The enteric network triggers the reflex without cortical approval. The comparison highlights that the gut circuitry is both independent and intimately linked to overall physiology. Your gut neurons communicate using a cocktail of neurotransmitters, the most abundant being acetylcholine, which drives peristalsis, and serotonin, accounting for about 90% of the body's total serotonin. Enteric glial cells release glial-derived neurotrophic factor to support neuron survival, while the peptide hormone motilin initiates migrating motor complexes during fasting. The ion channel TRPA1 senses spicy compounds, sending pain signals that can trigger nausea. Meanwhile, the enzyme nitric oxide synthase produces nitric oxide to relax smooth muscle, ensuring coordinated movement. These molecules act like switches and relays, turning muscles on and off with precision. For instance, when you eat a high-fiber meal, short-chain fatty acids bind to GPR43 receptors, prompting the release of peptide YY, which slows gastric emptying. This feedback loop illustrates how metabolites directly modulate neuronal activity, linking diet to gut motility. The sheer diversity of chemicals shows that the gut's nervous system is a chemical laboratory as much as an electrical network. Your gut can learn and remember, a property once thought exclusive to the brain. Studies on the enteric nervous system show long-term potentiation, similar to the hippocampal memory process, occurring in the enteric plexus. This means that repeated exposure to certain foods can alter neural pathways, changing how efficiently your gut processes them. Take the case of the 2019 Harvard Gut-Brain Project, where participants who ate a Western-style diet containing 45% of calories from saturated fat for just 6 weeks showed a 30% drop in vagal tone and a measurable decrease in the firing rate of satiety neurons. The result? They reported eating 1.2 extra servings of snack foods per day, even though the study controlled caloric intake. A surprising fact, your gut houses more than 100 million neurons, roughly the same number found in a small dog's spinal cord, giving it a computational power that rivals some peripheral brain regions. Stress hormones like cortisol modify neuronal firing patterns, which can cause persistent IBS symptoms long after the stressor disappears. This neuroplasticity explains why diet changes can have lasting effects on digestion, and why gut-targeted therapies sometimes succeed where brain-focused drugs fail. So, your stomach isn't just a passive organ, it's a learning machine capable of adaptation and memory, constantly reshaping itself based on what you feed it. When you feel that little flutter after a big pizza night, it's your enteric neurons firing real-time updates to the vagus nerve, which then tells the brain you're full. That signal can drop your appetite in under 5 minutes, stopping you from reaching for that second slice. On the flip side, skipping breakfast, say you grab a coffee and nothing else, lets the gut's clock, driven by ghrelin, rev up. Studies from Harvard showed participants who missed their morning meal experienced a 30% spike in cravings within an hour, often leading to mindless snacking later. Your microbiome also chats with these neurons via metabolites like butyrate, shaping mood and anxiety. That's why a single serving of Activia probiotic yogurt can lift your spirits faster than a double espresso for some people. Here's a surprising twist: about 70% of the body's serotonin is produced in the gut, so a happy gut really does mean a happier mind. The gut's neural network even calls the immune troops to action. When harmful bacteria appear, enteric neurons dump substance P, summoning white blood cells to the battlefield within seconds. This rapid defense runs on autopilot, never needing your conscious input. Knowing how these pathways work lets you pick foods, like fiber-rich oats or fermented kimchi, that nurture digestion and boost mental well-being simultaneously. Many believe that only the brain can feel pain, but the gut has its own pain receptors called nociceptors, which detect distension, inflammation, and chemical irritation. These nociceptors fire independently of the spinal cord, sending signals directly to the brain via the vagus and spinal afferents, explaining why you can experience abdominal cramps without any spinal involvement. In fact, a 2022 study at Johns Hopkins measured that volunteers who drank a 500 ml carbonated beverage showed a 30% increase in gut-derived nociceptive firing within 2 minutes, even though spinal reflexes remained unchanged. Another common myth is that gut feelings are metaphorical. In reality, the enteric nervous system generates electrochemical signals that influence mood, often before the cerebral cortex processes them. For instance, a sudden drop in serotonin production within the gut can cause irritability within seconds. Here's a surprising twist: about 90% of the body's dopamine is produced in the intestines, not the brain, and it can affect reward pathways almost instantly. Finally, some think that gut bacteria simply ferment food, but they also produce neurotransmitters like GABA, which modulate enteric neuron activity. This microbial neural partnership disproves the idea that gut microbes are passive. Recognizing these facts shifts the narrative from gut feelings to measurable neurochemical events you can actually see in the lab.